Good afternoon. Yeah, I can start if you prefer. So
Please.
Yeah, my name is Giulio Ruffini. Thank you for the invitation. My talk is about model-driven TS. Next slide, please. Okay, so we start from the technology, which is this multi-channel brain stimulation technology, and one of the challenges is to optimize it for something as complex as the human brain. Our goal is clinical, so we are very focused on helping patients in different pathologies, including epilepsy, Alzheimer, etc. in the company. Thank you. Next slide. You can skip this one. Next one. Yeah. So I'm going to be talking about neurotwins. So let me tell you what a neurotwin is in the context of my talk. So it's a compressed computational rendition of reality with sufficient detail and realism to optimize a brain stimulation protocol in the context of a specific pathology for a specific patient. So this means bringing together the physics of brain stimulation with computational neuroscience. Next slide, please. And here's a pictorial the description of the same that we use for explaining what we do with Alzheimer's disease. So we have data from patients from different modalities, from MRI, from different modalities of MRI, DMRI, fMRI, etc. PET, TMS, EG, whatnot. And the idea is to create a model, a computational model that replicates as best as possible the data in this particular patient and the pathology that they have. Okay. And with this platform, then we can go into the stimulation. Yes, next slide, please. So the approach to do this, of course, it's cannot be done brute force. So we have a multi-scale approach where we try to understand the effects of brain stimulation at the microscopic scale in the left, and then moving on to integrating the physics and physiology at the micro mesoscale, and then going to the whole brain model. And there are different brain models that one can use, from more abstract, such as the Ising model or the Hopf model, to very detailed mesoscale type of models with neuromass models and so on, which is what we call hybrid brain models, and it's the ultimate goal of what we're trying to do right now, is to create neurotwins with this type of framework. Okay, next slide. So physics. So this is what we have been doing so far. Next slide, please. Which is to integrate the physics, and one can start, of course, with the microscale, and we have this model in transcranial current stimulation that applying electric fields can modulate the membrane potential. From original concepts on this and models, we have now more detailed neuron models, such as the one that were pioneered by Aberra, and we have recently developed further this approach of working with compartment neuron models to understand really the impact of electric field on the membrane potential. Next slide, please. And yeah, basically we can now create models that give us a detailed representation of the effect of the electric field on the membrane potential of different types of models. And here at the bottom, I show you a list of interneurons. I mean, we of course have them for pyramidal cells, but we also have now models for interneurons, and this can help us understand the effects of the electric stimulation. Okay, next slide. The other thing that one needs to understand to go from microscale to mesoscale is this kind of surprising effects in TES, where even though the effects at the single cell level are very small, when you move to networks, you have an enhanced response that has physiological effects. And I think it's exciting now that there are coming out these new so-called next generation neuromass models that, as we recently showed, give a hint on why network effects in multiple cell models can give rise to enhanced electrical field stimulation effects. Okay, next slide. Yeah, so I guess everybody knows about the physics of modeling the macroscale electric field on the human brain using MRI data. This is something that we have now pretty much under control, I think. Next slide. Together with many others in the field. Next slide, please. Yeah, so we have now a pipeline in place where that we use for clients or for our clinical trials, where we can start from the MRI of a subject and build an electrical field model and then optimize the electrical field distribution based on a target specification, right? You may want to stimulate, like in this case, the left dorsolateral prefrontal cortex, and the algorithm gives you, with some constraints, what is the best electric distribution of electrodes in the cortex to do that. And this is operational now. Next slide, please. Yeah, so it has given rise to different studies. What I want to highlight, I think, in the next slides is what we're doing now in epilepsy. Next slide, please. In our epilepsy program, we have put in place this optimization method based on the physics of the electric field. Next slide. And we started with you can go through the slide until you show the full thing. Yeah. So we showed in a pilot study that using brain stimulation targeted into a target specified by a neurologist for refractory epilepsy patients could give to lead to a reduction on the seizure rate on these patients. And there are a couple of papers describing this now, and we are now working on a pivotal trial with this technology, with the physics technology applied to refractory focal epilepsy. Next slide, please. Okay, so now I want to talk about what the new things that we're doing in this regard, which are more research oriented. It's the physiology of neurotwins. It's trying to bring this computational neuroscience angle into the program. Next slide, please. And we start from the technology of neuromass models that has some history now. So we basically represent the activity of the brain by neuromass models, which are a set of coupled differential equations representing the dynamics of populations of neurons. And we have developed a modeling approach where we can now represent the interaction between fast oscillatory and slow oscillatory circuits and distribute them in the cortex. Next slide, please. And yeah, this we have some papers now describing how this can be applied in the context of macaque data on the left, with macaque data generated by MIT, and where we can show that we can actually replicate pretty nicely the depth profiles of power with these kind of models. On the right, you have a more recent paper where we apply this to SEG recordings in epilepsy patients, and where we seek to reproduce the seizure dynamics. Next slide, please. Again, here you can see the mix of physics and physiology because our models can represent LFPs and so on. Once we have this kind of neuromass modeling formalism that you can use in the for stimulation, you can ask what is the resonance properties, for example, what is the response to electrical field stimulation in TES, which is what we're interested in. So we have TDCS, our ACS, and here you have an example of the Arnold tongues for the two pyramidal cell populations in this particular model. Next slide, please. Yeah, okay, so a bit more, yeah. So as I mentioned, we can also personalize models for patients in epilepsy to represent pathologies having to do with KCC2 transport dysregulation, which we think is important to then better define the brain stimulation protocols for these patients. And you have a link there to a preprint if you're interested. Next slide. We are working now on Alzheimer's disease, and the first step to do that has been to try to understand as best as possible the etiology of Alzheimer's and the representation, the mechanistic representation of pathology in our neuromass models. So here you have in the slide on the left a pictorial representation of what we understand are the causes and the mechanistic effects of Alzheimer's disease from original inflammation due to pollution or infection, to beta amyloid, to the failure of PV cells and hyperexcitability. And we can represent this now, I think, in this kind of neuromass models as well, and we can use these pathologically representative models to better understand how to stimulate them. Next slide. The Microscale, next slide. Well, the basic idea is to ingest. So once we have these kernel neuromass models, we parcellate the brain, we allocate the models to each parcel, and then the task is to create a whole brain model. And for that, we typically ingest things like MRI for the physics part, anatomical MRI, then functional MRI, DMRI for the connectome, and then if we have EEG or SEG and PET, these are all things that the models can try to represent to construct a whole brain model that is what then ideally we can use to best define the target of stimulation, because once we have a definition of the problem in the model, we can actually define the target. Rather than saying stimulate here, we can say stimulate so that you increase, for example, gamma power in the brain, and things like that. Okay, so this is kind of the overall picture. In order to fit models, then we have to look at the data in different ways. Like with fMRI, we can construct functional connectivity maps that the models need to reproduce, or dynamical functional connectivity features. And here we're working, we're fortunate to work with people here in Barcelona, Gustavo Deco's lab, who is a leader in this field. Next slide. And yeah, once the models are constructed for a particular patient, once the neurotwin is ready, then we can do the stimulation and do the nice stimulation. Here you have kind of it's a bit of conceptual because it's a very early simulation, but what happens when you apply pathology to a healthy neurotwin, you apply pathology representing Alzheimer's disease, what happens to when you apply brain stimulation to this patient, and the idea is to try to recover the dynamics that are present in the healthy model. Next slide, please. Yeah, so this is kind of a bit of the summary of where I think we are going. So we started with what they call TES 1.0, where we work with sponges, with the target definition based on the 1020 system, without really any definition of a brain modular optimization function, to where we are now in our clinical trials with small electrodes and physics-based targeting from targets specified by neurologists typically, and where we focus on the electric field, to what the last step is, where we want to actually have the target functions being defined by dynamics as well. This is my team in Barcelona, who I'm very fortunate to work with. And in this next slide, you have a list of collaborators whom I wish to thank. And thank you all for your attention.
Thank you very much. Thank you very much.